Hello students. Today we are going to learn about one of the fundamental reactions of photosynthesis. Okay, now photosynthesis, which results in the plants being able to synthesize glucose, is composed of two reactions. One is known as the light dependent reactions, the other is known as the light independent reactions. Now, light dependent reactions are one that take the source of the energy required for those reactions from the light, okay, and it results in the synthesis of two products, ATP and NADPH. So light dependent reactions results in the synthesis of ATP and NADPH. Okay. Basically NADPH plus H plus. These two products are utilized in the synthesis of glucose in the light independent reactions. They are utilized for the synthesis of glucose, which results in the synthesis of glucose for the target molecule of photosynthesis. Okay. Now, I am assuming that whoever will see this video, if a student sees this video, you already know about the photosystems. Photosystems are what? They are composed of the light harvesting complex molecules as well as electron acceptors. Chemiosmotic hypothesis is the mechanism which explains that how chloroplast is able to synthesize ATP and NADPH. Alright. So, where actually this reaction is taking place? It is taking place on the membrane of the thylakoids. Okay. It is taking place on the membrane of the thylakoids. Now, in order to understand the site of this reaction, a little bit of revision of that chloroplast structure is essential. All three together are known as grana. A single tower is known as grana. The base material or the you can say the medium is known as the stroma. And these 5 rupee coin like structures that you are seeing, each of them are individually called as thylakoids. Of course, these bridges that you are seeing, they are known as the stroma lamellae. But currently we are not focusing on the reactions of stroma lamellae. We are focusing on the reactions that will take place in the membrane of the thylakoids. Okay, so I will just take one thylakoid and I will zoom into the structure. A single thylakoid will look like this. It has a single membrane. I am using this as the lower side of the membrane and upper side of the membrane. It is not double membrane. Okay. Now, in this part, this side is the inside the volume enclosed by the thylakoid is known as the lumen, while outside the thylakoid of course is the stroma. Okay. Now, since you have read a little bit of the non-cyclic photophosphorylation, you know that electrons are passed from the PS2 first. So, but that was just a schematic diagram. What is actually the reaction? Actual reaction takes place here. This is the PS2. PS2 releases the electron first. It moves to an electron acceptor. That electron acceptor passes the electron to a molecule, an electron carrier. It's not an electron carrier, but for the sake of now understanding, we will say that the electron is passed to a molecule that is plastoquinone, which then passes it to cytochrome B6F, then it passes to another molecule, plastocyanine, which then passes to PS1. Collectively, these are the three molecules together which you have already, you call it as electron transport system. Electron transport system. And finally, on this side there is other molecules that are associated, and they are responsible for the synthesis of NADPH. Okay. Now, here, what happens here is that this is the whole number of molecules that are total present on a thylakoid membrane. As soon as the PS2 gets excited and leaves the electron, it is without any electron. Okay. So we need to understand here two things. One is, I'll come back to this picture again. 2 points need to be very important. One is the pathway of the electrons. Okay. Next is how that electron movement leads to building up of a proton gradient. Now, what is proton gradient? See, any gradient means difference. So it is basically proton concentration difference across the membrane of the thylakoid. Across the membrane of thylakoid. Okay, across the membrane of the thylakoid. That is, inside lumen more protons, more H plus, and in stroma it is less. Alright. And the last part is how this proton gradient is responsible for synthesis of ATP. For ATP synthesis. These are the three crux points of this whole chemiosmotic hypothesis, the mechanism that is responsible for the synthesis of ATP. Okay, now coming back here again. See, PS2 is the first photosystem, the LHC of PS2, the chlorophyll A molecule of the LHC of PS2 is the first molecule that releases the electron. As the electron moves to the electron acceptor, PS2 cannot remain in an extended condition of positively charged state. It then itself will bind with oxygen because oxygen is so electronegative. It then itself will bind with the oxygen and will form a chlorophyll oxide, which will stop the reaction, because the whole reaction is dependent on the fact that the chlorophyll molecule goes on to release the electrons. So PS2 has to ensure that a fresh supply of electron is coming to it. How it ensures that? It carries out the photolysis of water. It carries out the photolysis of water, leading to the formation of this. Okay, oxygen is the byproduct, goes out of the stomata. This remains here. The first reason that the proton concentration is building up inside. Electrons are taken up, electrons are passed to the electron acceptor, and the electron acceptor, as soon as it passes the electrons to plastoquinone, the problem with plastoquinone is not a electron carrier. Plastoquinone is a hydrogen carrier. As it is given an electron, it will pick up a hydrogen ion, that is a proton, from the stroma side. And it will temporarily become EQH2 plus 2 quinol, and again it will become EQ plus H2. Now the problem is that PQ tries to pass the H2 to this molecule cytochrome B6F. Cytochrome B6F will not take H plus. Cytochrome B6F will not take H plus. Cytochrome B6F will do what? Cytochrome B6F will take the electrons. Okay, so therefore what will happen is that this process will not take place. Cytochrome B6F will take the electrons only, ionize this H2 molecule, and will do what? Will push the protons inside. So that is number 2 of why the concentration of protons is increasing inside the lumen. Okay. Third most important thing is that as this electron is passed to plastocyanine, that is passed to PS1, PS1 takes up the electron with the help of these two enzymes, ferredoxin. It's not an enzyme, it's a compound that is present there, and FNR, ferredoxin NADP reductase, it is an enzyme. With the help of that, it will synthesize NADPH plus H plus. Where will this H plus come from? The third reason of the building up of the proton gradient is that NADP plus is so electropositive. The NADP plus, when it is given electron, it will pick up protons from the stroma side, and it will ultimately stop in a configuration like this. So these two extra protons that the NADP is again pulling from the stroma side will also contribute to increasing the difference between the lumen and the stroma. Inside the lumen there will be too much of hydrogen ion, inside the or outside the lumen, that is in the stroma, there will be too little of hydrogen ions. Okay, so this is the story of the building of the proton gradient. Now see, increasing the proton gradient does two things. One, inside this whole space, the lumen, it becomes acidic in nature, the pH constantly goes down. Third thing is, you are accumulating proton, that means you are accumulating charge. So between the inside of the thylakoid and outside of the thylakoid, there will also be charge difference. There is going to be electrochemical gradient or charge difference, and as a result what will happen? Since there will be charge difference, there will be a potential difference. So after a point, there is going to be a breakdown of this charge. There is going to be a breakdown of this charge, and H plus ions will start to go out. Okay. This is the ATP synthase molecule. This is the ATP synthase molecule. It has got two parts, CF0 and CF1, complex F0 and complex F1. Okay. So complex F0 will capture in the will allow the passage of the H plus ions to move, and also it will get the kinetic energy from the H plus ions to move when it is moving outside. Complex F1, when it receives this energy from this CF0 molecule, complex F0 molecule, its configuration changes, and CF1 does the important thing. CF1 uses this energy to bind ADP and inorganic phosphate plus energy to form the most important energy currency of the cell, that is adenosine triphosphate or the ATP molecule, which was basically the goal. So one goal was done by PS1 associated with FD and FNR. They synthesized NADPH plus H plus. The other goal was done by CF1, which is part of the ATP synthase complex. CF1, CF0, they make up the ATP synthase complex. Among them, CF1 changes its configuration, binds with ATP and PI, uses the energy it receives from CF0, CF0 which receives the energy from the passing out of the hydrogen ions, and finally synthesizes the ATP molecule. Alright, that's it. Hope you have understood a little bit, okay, about chemiosmotic hypothesis. Bye.